Hi, everyone. Thank you so much for that awesome introduction. And thank you so much again to Thermo Fisher for allowing me to be part of this consortium of amazing researchers, which is the Gibco Cell Culture Heroes. And thank you also for the opportunity to come and give you this presentation. So I thought I would just start with a little bit of my academic background. And I think anybody who would be looking at it would probably just think that this guy must really like Birmingham because I've spent most of my academic time here. I completed my undergraduate master's, which is an MSci in biological sciences at the School of Biosciences at the University of Birmingham in 2008 to 2012. I then came back in 2013 to complete an MRes leg of my 1+3 PhD program, which was based in the Institute of Immunity and Infection. And this was a really great program because it just gave me this opportunity to do different projects in the Institute for Biomedical Research at the University of Birmingham and really figure out what I wanted to do for my PhD. After this, I settled on a PhD at the Centre for Liver and Gastrointestinal Research, which is based on the 5th floor of the IBR. And following this, I settled in the CLGR and continued my postdoctoral research fellowship. Now, it is true that I do really like Birmingham, but one of the main reasons I stayed is just because of how good Birmingham is for the study of the liver. Now, before my MRes, I didn't really know much about the liver, but in time, I kind of realized that it is amazing. It's the largest internal organ with over 500 different functions and composed of around 80% hepatocytes, which perform most of these functions. This includes manufacturing bile in the aid of fat digestion. They're involved in processing digested food from the intestine, controlling levels of fat and amino acids and glucose in the blood, and also involved in neutralizing and destroying all drugs and toxins. Now, as important as it is to understand what the liver does under normal circumstances, it's even more important to try and understand what happens when it goes wrong, in the case especially of chronic liver disease. Now, this is quite poignant for the UK because, as you can see from this rather scary graph, liver disease is the only cause of death in the UK that has actually been increasing in incidence in the last 50 years. So this is a really important burden of this disease in the UK and a very large requirement for us to try and understand chronic liver disease and therefore provide new treatment avenues. Now, in Birmingham, we are very good at trying to do this because of a long collaboration with this building here, which is the Queen Elizabeth Hospital. This is the largest solid organ transplant centre in Europe and a national specialist in liver transplantation, performing 150 to 200 liver surgeries a year. Also, it possesses a 20-year-old program with a CLGR, which grants us this unique and precious access to excess surgical tissue and blood from patients with liver disease. But I think what's more important as well, not just with this collaboration, is that the IBR lives about a 5-minute walk away. So this means that just after a single phone call, we can have access to authentic diseased human tissue within 5 minutes, which means we can use this resource in its freshest possible format. So we are in prime position to study human liver disease. Now, these advantages have given rise to my research program in the past, which I shall hopefully give you an overview in this presentation. So I will talk about liver cell and cell structures and this process known as enclysis. I will then talk about ex vivo imaging of liver tissue with multiphoton microscopy, and then I'll finally talk about high content imaging and CD8 positive T cell internalization by biliary epithelial cells. So just to start off, it's important to really understand what cell structures are. Well, for most of you, I think they're exactly what you would think they are. They are the presence of one cell inside another cell. This generally arises from the activity of Rho GTPases, which allows one cell to invade the other. This leads to the presence of one cell within a vesicle within an actual host cell. Now, there can be various outcomes for this internalized cell. It can either be released, leading back to the generation of single cells. The internalized cell may thrive inside the host cell and divide, or it could be deleted by its host cell by lysosomal fusion. Now, to try and understand what the implications are of cell and cell structures for the liver, you first need to understand what the cellular architecture is of the liver. Generally speaking, the liver is composed of these hexagonal subunits, which are known as lobules, which you can see on the left. At the corner of these lobules is a collection of smaller ducts, which are split into the portal vein, the bile duct, and the hepatic artery. And when you zoom in On the tracks of the portal triads, you can see that blood derived from the portal vein and the hepatic artery mixes within small capillary-like structures, which are known as sinusoids, which then travel collectively to the center of the lobule into a larger structure called a central vein. These sinusoids are lined with endothelial cells, and which also associate with a resident pericyte population in the liver, which are called stellate cells, as well as a resident macrophage population, which are called Kupffer cells. Outside of the sinusoids are the hepatocytes, which also form small vessels called bile canaliculi, which transport bile in the opposite direction collectively towards the bile ducts, which are lined with biliary epithelial cells, which I'll talk a bit more about, and I'll talk a bit more about these later on. Now, as we've touched upon, the main cell type of the liver is the hepatocyte. So it's no surprise that most of the research into cell and cell structures has been largely focused on hepatocytes. And already there are 4 main forms of cell and cell structure associated with hepatocytes. On the right, you can see a detail of suicidal M. peripolesis. This is the internalization of autoreactive CD8 positive T cells by hepatocytes, which are then subsequently deleted. On the left, there is a process known as mitosis, which often is a cancer-associated process and involves the heterotypic internalization of one cancer cell by another. And this is associated with interactions between beta-catenin and E-cadherin. But for the sake of this talk and what my PhD was focused on, I'll talk about these 2 processes. At the top is a process called efferocytosis, which is not unique to the liver, but is performed in association with ASGR1, or asialoglycoprotein receptor. And at the bottom was the main focus of my PhD, which is a process which came to be known as enclysis, which is the internalization of live CD4 positive T cells. I'll talk a bit more about that now. So the main focus of my PhD was the biology of cell and cell structures in the human liver, which largely involved comparing this process called enclysis to efferocytosis. Now, at first, we didn't really know that much about it, so we decided to start looking ultrastructurally using scanning electron microscopy, which is shown here. And we found that phenotypically these processes were quite different. Enclysis involves the direct generation of blebs on the membrane of hepatocytes, or HOH7s, which is a hepatoma cell line that we used for our assays. And we can see that they're reaching up around the live CD4 positive T cell to gradually engulf it. However, with efferocytosis, you can see the generation of membrane ruffles, which are reaching up to grab internalized dead cells. So this already gave us an implication that these processes were different and separate. To further understand as to whether this process was actually unique to CD4 positive T cells, our early work was focused on co-cultures between different hepatocyte cell lines or primary human hepatocytes with either CD4 positive T cells, CD8 positive T cells, or CD20 positive B cells. And what we found in the case of CD8 positive T cells and B cells was that regardless of what hepatocyte cell type we used, these cells tend to just travel in between the gaps of the hepatocyte cells or intermingle within monolayers. However, CD4 positive T cells generated these large internalized vesicles where they resided, which would imply that they were being internalized. Following this, we decided to try and understand what the molecular mechanisms were of this process. Generally speaking, the recruitment of immune cells is associated with the use of adhesion molecules, such as intercellular adhesion molecule 1 or ICAM1. So we first tried to see if these adhesion molecules were involved in this process. By performing immunostaining, as you can see on the left, we can see that ICAM enriched around CD4 positive T cells, which were attached to the surface of HOH7s. Additionally, using antibody blockade, we realized that anti-ICAM1 antibodies were able to significantly reduce the amount of internalization of CD4 positive T cells. However, as shown on the column which says enclysis, we saw that cells which had already been internalized did not associate with ICAM1. So this told us that ICAM1 was largely associated with the adhesion step of this process. So we wanted to try and understand what was actually involved with the internalization process itself. As I mentioned about mitosis, there are certain molecules which can be associated specifically with internalization, one of which is beta-catenin. And again, by performing immunostaining and then performing 3D volume reconstructions of Z stacks, we can see that internalized CD4 positive T cells are enriched for beta-catenin. And to try and make sure that this was actually a real observation for enclysis and not just an artifact of cell culture, we decided to perform similar staining using immunohistochemistry. With this, we actually stained tissue sections which were cut thicker than usual to try and include to make sure that we were able to capture the entirety of an internalized cell. And what we found by staining for beta-catenin and also a transcription factor for an infected T cell, which here is T-bet, we can see that again an internalized T cell does indeed associate with beta-catenin, which showed that what we saw in vitro was indeed representative of what happens in the tissue. So we were able to show that beta-catenin was involved. But what about E-caterin, which is the other protein associated with entosis? We already knew that hepatocytes were capable of entosis by looking at our own hepatomal co-cultures. And what we can see that hepatocytes can actually internalize their neighbors and put them into vesicles, which are enriched for E-caterin. However, when we performed the same staining for cells which engulf CD4 positive T cells, we saw that the vesicle containing the T cell did not enrich for E-caterin. Additionally, when trying to block these processes using published inhibitors of entosis, which are shown here, which are Y27632 and H1152, which are inhibitors of Rho kinases, we can see that there was no effect. So this showed us that this process was indeed not related to entosis. So we can see that this process is unique and unrelated to other forms of cell structures, and we know that it specifically involves CD4 positive T cells. But CD4 positive T cells encompasses a wide variety of different subtypes with different biological roles. They can be broadly split into regulatory T cells, or Tregs, and non-Tregs, or T effector cells. Tregs are anti-inflammatory, and they act by secreting anti-inflammatory cytokines such as IL-10 and TGF-beta. And these particular cells are very largely abundant in the liver. Non-Tregs, or T effector cells, can be subdivided into different types of pro-inflammatory cells, but for the case of the liver, they are largely abundant with Th1 type cells, which cause inflammation by secretion of pro-inflammatory cytokines, which include interferon gamma and TNF alpha. So due to these differences, we were wondering if, following their internalization, were these cells actually treated any differently by the capturing hepatocyte? Well, when we perform co-cultures with Tregs and non-Tregs in the presence of fluorescent reporters of acidification, we found that Tregs were more likely to enter acidic compartments compared to non-Tregs. This implied that enclysis was actually a method in which hepatocytes were actually deleting regulatory T cells. Again, we wanted to see if this was the same case in tissues. To do this, we performed semi-quantitative analysis of donor liver tissues and also tissues from patients with a wide variety of chronic liver diseases. And with this, we quantified the amount of cells expressing FOXP3, which is a Treg marker, or T-bet, which is a Th1 marker. And what we found, is that in all cases, the number of cells expressing FOXP3 found in the cytoplasmic spaces of hepatocytes were always higher compared to those expressing T-bet. Moreover, what we also noticed that this difference was a lot more exaggerated in a disease called autoimmune hepatitis, or AIH. This disease is a specific targeting of hepatocytes by the immune system and is an autoimmune condition. So with this in mind, it could imply that cell-to-cell structures may play quite a wide variety of roles in chronic liver disease. By the nature of suicidal peripoiesis, so we already know that it's associated with autoimmunity, as it's only CD8 positive T cells that are actually engulfed by hepatocytes in a mouse model. Erythrocytosis also can contribute to disease if dysregulated. And as we've already shown, if Tregs are deleted by enclysis, which might lead to uncontrolled inflammation due to the reduced number of Tregs. So generally speaking, this shows that cell structures, may play quite a large number of roles in chronic liver disease. So just to summarize this part, the liver is a hotspot for cell structures. Tregs are preferentially deleted by hepatocytes through enclysis, and enclysis itself may contribute to chronic liver disease pathogenesis, especially of autoimmune hepatitis. Now, at this part, it might be clear now that combining in vitro observations with in vivo observations is a powerful tool to show how rigorous your observations are. However, the goal it'd be much more appealing to be able to actually study cell and cell processes in live human tissue, because so far what we've shown is largely just a static readout because of the nature of staining. So ideally, it would be nice to actually answer the question of what is really happening in tissues in terms of cell and cell structures. Well, this kind of became the main basis for my first postdoctoral post. This project was entitled Multiphoton Imaging in Human Liver Tissues, Validation of a New Tool for Drug Discovery. Now, this was a training fellowship funded by the National Centre for the Replacement, Refinement and Reduction of Animals in Research, and aimed to develop a model using multiphoton microscopy to provide a means to reduce or replace the use of animals for intravital microscopy of the liver. So why would you use intravital microscopy to image the liver? Well, many scientists use mouse models to actually study chronic liver disease. This can include the administration of acetaminophen or paracetamol, the administration of a high-fat diet, or by the use of concanavalin A. These provide simulations for hepatotoxicity, dietary problems, and inflammation, respectively. And combining this with intravital microscopy, where the liver is exposed in an anesthetized animal, you can observe a bit of the activity of the cells in a live animal. However, these aren't necessarily good representations of human liver disease. So if you have actually access to authentic diseased human tissue, why not just image diseased human tissue instead? So this formed the main question of my fellowship, and we decided to approach this using multiphoton microscopy. And the reason we decided this is because it has a lot more benefits to using multiphoton microscopy compared to conventional one-photon microscopy. So the differences are that with one photon microscopy, as shown on the left, a single photon is used to excite your sample, and everything in the pathway of that photon is excited. However, with multiphoton microscopy, this involves the use of two or more low power beams which meet in a single spot. And when these beams meet near simultaneously, this quadratic effect happens, which suddenly causes an increase in power and therefore excitation in a single spot where these lasers meet. This means that you can image your tissue much deeper, and it also means that you're causing less damage to the sample as you're only imaging one spot, which makes it perfect for imaging live tissue. So with this in mind, we generate a brand new model whereby pieces of liver tissue are sat in a 6-fold plate using custom 3D printed inserts and are placed directly under the objective of our multiphoton. This tissue is then connected to a peristaltic pump, which provides perfusion. This peristaltic pump is connected to an inline heater as well to make sure that the sample is kept at normal thermic conditions. And this means that the tissue can be imaged and perfused at the same time. So our first question was to prove that the results that we could obtain using this system were actually comparable to that of intravital microscopy. To do this, we actually use ex vivo mouse tissue at first with the system to compare it to intravital microscopy. Now, with the use of fluorescent dyes, you can light up the sinusoids of the liver and also identify immune cells that were patrolling them, which is shown here on the left. Using a slightly different combination of dyes with our ex vivo model, you again can see that we can see the sinusoids as well as the immune cells that are associated with them, but also you can see the nuclei of the hepatocytes, which is shown here in white. So this shows that we can actually get the same quality and same detail of images using this technique, which you can get from intravital microscopy. Of course, one of the main benefits as well of using intravital microscopy is that you're looking at viable tissue. So we also need to prove that our ex vivo tissue was also indeed viable. To do this, we labeled our tissue using fluorescent reporters of active mitochondria. In this case, it was mitotracker deep red. And the reason we use this is because this dye only fluoresces if the mitochondria are active, and therefore if the cells are alive. And what we can see here is that when we do this, we can see that the hepatocytes indeed show active mitochondria, and showing that our tissue are indeed alive when using this system. So far, this shows that this model is comparable to intravital microscopy in terms of the quality of information that you can get, and also because the tissue that you're looking at is indeed alive. So our next step, after we validate this model, was to try and use it to study cell and cell structures. To do this, we initially focused on studying efferocytosis. To do this, we induced a burn injury into a piece of tissue. with the logic that the viable tissues around the dead cells that we've induced with a burn injury would be able to clear away the dead cells and therefore perform phagocytosis, which we can then image using multivector microscopy. To be able to see these cells, we labeled our tissue with an impermeable dye which shows dead cells, which is shown in dark blue, as well as using a general dye of all cells, which is in this case is cell tracker green, and our usual membrane and nuclei dyes. And what we can see is not only can we identify the actual position of the burn injury, but if we look deeper in 3D, we can identify where a hepatocyte has been internalized by its neighbor. So this shows that this model can indeed be used to study cell and cell structures in real time. So just to summarize this part, we have shown that both human and mouse liver tissue can be imaged ex vivo while also undergoing passive perfusion, and also that cell and cell structures in live human liver tissue can be imaged in 3D. Now, following this, the next steps were to finally observe cell and cell structures in real time in real diseased human liver tissue. But like with a lot of different projects, this guy had other ideas. Now, unfortunately, because of the COVID pandemic, our access to human liver tissue was very much ceased, so we weren't able to progress this project as much as we have liked. However, with the lockdown came a few more benefits and more opportunities to learn new techniques. So after a brief time of lockdown, I came back to the university and I was able to learn how to infect Vero cells, which at the time were the gold standard for infections for SARS-CoV-2. And I learned how to infect these cells in HSC class 3 cabinet conditions with the idea of developing an assay which we could use to test potential therapeutics. However, we didn't really have an option as to how we could test a large number of therapeutics at one time. So with this, we aimed to develop a high content imaging assay to test strategies for preventing SARS-CoV-2 infection. So what do I mean by high content assays or high content imaging? Well, these are large scale automated imaging acquisition and processing platforms which allow for unbiased quantitative data accumulation for a large number of images. So compared to normal microscopy, as I've shown this diagram here, you may have to look at your wells one by one and take different images which may not necessarily be in the same position, and it takes a very long time, and it gets to the point that you might not want to be able to, you might not have time to actually image your entire plate, as I also didn't have time to put crosses in all of the wells that you see here. However, in terms of using high content imaging, you have an automated platform which sequentially images all your wells of interest in the exact way that you would like them to be sampled with inside your well. So as you can imagine, this comes with many other benefits. You get a lot of time saved from repetitive imaging experiments where you ordinarily would have to image every well one by one. It's a high throughput platform which allows for simultaneous experiments to be performed in single plates, and it reduces the requirements for cells and reagents because you're using a much smaller volume of cells for each well. There's reduced human input for most cases, and the automated analysis allows for large amounts of data to be collected. So with this, you have to also ask yourself as to how you would analyze so much imaging data that's generated using these platforms. Well, there's not really one right way to do it, but for the case of this SARS-CoV-2 infection model, we decided to use a Thermo Fisher Cell Insight CX5 high content screening platform. And with this, we were able to develop an algorithm which actually quantified infected cells. And the benefit about using the CX5 is that it actually images but also analyzes your data at the same time, which means that once it's finished, you have both numbers and your images. And we managed to use this quite effectively to test a few important therapeutics that were being tested in Birmingham. One of which was a nasal spray that was carrageenan based, which was theorized to be able to prevent the infection of SARS-CoV-2. When you look on the images on the left, where you can see this lawn of Vero cells, you can see that with even going up to almost 1 in 300 dilution of this carrageenan-based nasal spray, we can see that it was effective in preventing these cells from being infected with SARS-CoV-2, which is stained in green here for the spike protein. And when we look at the quantification of this, we found that carrageenan-based nasal sprays pretty much prevented any infection of these cells. And because of the success of this publication, this nasal spray has now been actually put into production, as shown here. Furthermore, we also managed to provide some data which showed that drugs which control the blood levels of fat, which included phenylfibrate, were also able to reduce the infection and spread of SARS-CoV-2, as shown here. So once all this work was completed and we got a little bit more normality, we then went back to cell structures. At this point, we then asked the question as to whether we can actually analyze cell structures using high content imaging. So for this, I actually turned my attention to a different form of liver cell structure. And what this was the fact that CD8 positive T cells were shown to actually form cell structures within biliary epithelial cells, or BEC. What you can see here is an immunohistochemistry staining, which shows quite a large number of CD8 positive T cells within the cytoplasmic spaces of biliary epithelial cells, which are identified by cytokeratin 19 staining here. So, as beforehand, we didn't really know how we were supposed to study this. So the first job was to develop an in vitro model to then study this, as we did previously with Enclysis. So with our very privileged access to explanted liver tissue from the Queen Elizabeth Hospital, we used a protocol which combines enzymatic digestion and gradient separation to separate the epithelial cells from primary human liver tissue. With this, we use anti-EpCAM magnetic isolation to isolate the BEC and then put them into culture. With this, we co-cultured them with CD8 positive T cells, which were isolated using gradient density isolation methods from blood from patients with hemochromatosis, again at the Queen Elizabeth Hospital. These were then activated prior to co-culture. So again, because we didn't really know a lot about the process, we started off by looking ultrastructurally using scanning electron microscopy. And what we saw is that they were very different to what we previously saw with Enclysis. When the cells attach, we can see that the CD8 positive T cells seem to spread out across the surface of the BEC and form lots of individual contacts between the BEC and seem to be tugging at the actual membrane itself. Also, we found that in some cases, which is shown here on the right, that these CD8 positive T cells would polarize and then look like that they were actually starting to breach the membrane. But what was more striking, that compared to other forms of cell structure formation, the actual host cell, which in this case being the BEC, didn't seem to form any new membrane structures. So when you compare that to Enclysis, for example, where we saw the blebs reaching out to internalize a CD4 positive T cell, we saw that the BEC was a lot more of a bystander here, and this would imply that the CD8 positive T cell was invading the BEC. This also implied to us that this is probably quite a different process to Enclysis, and that also gave us a bit of permission to give it a new name. In this case, we decided to call it bectosis instead. So we went back to our tissue to try and understand if this was actually specific to CD8 positive T cells. To do this, we performed immunohistochemistry staining for CD8 and CD4 in our liver patient samples. And what we found was not only that these CD8 T cells that were invading seemed to be larger compared to the cells that were surrounding the bile ducts, but we saw that despite that the number of CD4 positive T cells associated with bile ducts far outweighed the numbers of CD8 positive T cells, they didn't look like that they were actually entering the biliary epithelial cells, and instead were just finding gaps within the biliary epithelium itself. So this gave us an idea that this was indeed a new process and specific to CD8 positive T cells. So the next question following this was to try and see if we could recapitulate this in vitro using the model we developed earlier. With this, we then developed a high content imaging assay to try and understand the molecular mechanisms of this process. To do this, we performed co-cultures with BEC that were previously labeled with cell tracker green with activated CD8 positive T cells, which were alternatively labeled with cell tracker red. They were then co-cultured for 4 hours and then labeled using a membrane stain. In this case, we used wheat germ agglutinin conjugated to Lexaflor 680, which we bought from Thermo Fisher. We then fixed the cells and then analyzed them using high content imaging. And when you use this strategy, you can take advantage of the properties of these labels to actually develop a high content imaging analysis platform. When a cell is internalized, it displaces the cytoplasm of its host cell, which you can see by a black hole in the BEC channel, which you can see on the left here. And this corresponds to the localization of a red CD8 positive T cell in the same place. Also, what's more is that when you label these cells with a membrane label at the end of the experiment, if a cell is internalized, it will actually be shielded from this membrane label, and which you can therefore confirm that the cell is fully internalized. Using the cytoplasmic displacement and the lack of the membrane staining, we developed an algorithm which would actually quantify cell structures using Cell Profiler. And what we found that the time post-activation of these CD8 positive T cells had an impact for the frequency of these cells internalizing into Beck. What we found that compared to 24 hours post-activation, cells that were activated 40 hours before co-culture were actually much larger than those that were activated for 24 hours. But almost counterintuitively, the larger cells seem to invade the Beck more frequently than the smaller cells that would have been activated for 24 hours, which is shown here on the right. But what's more, this kind of gave us this implication that we're recapitulating what we saw in the tissue. However, what was not clear yet was what was so special about these larger cells activated for 48 hours, which encouraged them to actually be internalized into these cells. So to try and begin and answer that question, we performed phenotyping using flow cytometry. And what we found that what we saw with flow cytometry looking at the forward scatter was that between 24 hours and 40 hours, there's this sudden shift in size, which is shown here by forward scatter. So this again confirmed what we saw with our imaging platform. But upon further analysis, we can see that these same 48-hour activated cells suddenly express a lot more of an integrin known as CD103. Moreover, these same cells express more of that activation marker, which is called CD69. So once again, we wanted to relate these observations that we saw in vitro to what we were seeing in vivo with our tissue staining. Therefore, to confirm our observations in vitro with what we were seeing in tissue, we performed multiplex immunohistochemistry, and what we found by staining for CD8, CD69, and CD103, we found that the same CD8 positive T cells that were invading these biliary epithelium, as shown here by Ikaterin staining, were indeed expressing the same markers that we saw using flow cytometry for cells we generated in vitro. And to confirm that these cells were indeed fully internalized within the biliary epithelium, we again performed immunohistochemistry staining for a thicker tissue section, which would encapsulate the entirety of an internalized cell. And what we can see here, shown by the white arrow in this 3 reconstruction on the right, we can see that a CD103 positive CD8 T cell is indeed fully enclosed within Ikaterin, and CK19 staining, which belongs to a biliary epithelium, confirming that these cells are indeed fully internalized. So finally, I will summarize this part and say that high content imaging was unveiled how large eccentric CD69, CD103 positive, CD8 positive T cells can invade biliary epithelial cells. And I've shown that combining in vivo and high content imaging in vitro experiments is important for cross-validation of observations. And if you'd like to learn more about this form of cell and cell structure, and especially how these CD8 positive T cells can also express Ikaterin, we've actually managed to publish this as a preprint in Research Square, which is shown here at the bottom. So please go and check that out. So, as an overall summary, high content imaging with automated analysis allows rapid and multi-parametric confirmation of in vivo observations. The frequency of cell and cell structures is influenced by human liver disease and likely vice versa. And our observations made in vitro, in vivo, and ex vivo using different and appropriate forms of microscopy provides more rigorous findings. So with that, I'd just like to acknowledge a few people. These things get larger and larger the more you go through your career. So I don't really have time to thank everybody individually, and I'm sorry if I've missed some people, but I would just like to thank my supervisor, Professor Yehu, for giving me the freedom to carry on working with cell and cell structures, and also particularly Dr. Vincenzo Ronca, who's been really helpful and really invested with the biliary cell and cell work. And finally, I'd also like to thank Dr. Zanya Stamataki and everyone who's been in our team beforehand for all of their help with the work on in crisis and allowing me to work on COVID during the pandemic as well. And thank all of you for coming, and I'll take any questions. Thank you all so much.